audible to all? Please write yes if I'm audible. Okay. Am I audible to all? Okay, 1 minute. Start our webinar. So, hello everyone. I am Somnath Dash, Chairman of TIOSCASS, the Institute of Science, Culture and Social Studies, and also the CEO of SDLIFESCIENCE. Today we are going to conduct the webinar on therapies and research on the COVID-19. So I'll not take so much time. Okay, so I today two eminent speakers. Yeah, today two eminent speakers will give their lecture. Okay, so without wasting any time, I would like to introduce our first speaker, Dr. Dipankar Ghosh. Okay, so the session is handed over to Priyanka. Priyanka, please start. Hello, Priyanka. Hello. Hello. Yes, Priyanka, you can start. Yeah, you can start. প্রিয়াঙ্কা মাইকটা অন করতে হবে প্রিয়াঙ্কা মাইকটা অন করতে হবে
Hello everyone, good evening. This is Priyanka Talukdar, Secretary and Managing Director of SDLIFESCIENCE and TIOSCASS. I would like to introduce Dr. Dipankar Ghosh. He is an Assistant Professor and a Microbiologist in Department of Biotechnology at JS University, Kolkata. He has completed PhD, MPhil and MSc in Microbiology from University of Montreal, Canada, Annamalai University, India and Calcutta University, India, respectively. He has conducted research work as postdoctoral research associate and research fellow with individual fellowships in different prestigious organizations like the University of Cambridge, UK, Imperial College London, UK, Pasteur Institute of Paris, France, McGill University, Canada, Pusan University, South Korea, University of Turku, Finland, and IIT Kharagpur for diverse academic and research purposes. He has already published more than 100 peer-reviewed high-impact research articles, book chapters, and conference preceding time to time. He is currently running microbial engineering and algal biotechnology laboratory in Department of Biotechnology, JS University, Kolkata. He is currently supervising 4 active PhD research scholars, 16 MSc research project trainees, 23 BSc project students, and 1 BSc external summer internship student respectively. At present, Dr. Ghosh has 2 industries-funded research projects to support his research activities in Department of Biotechnology from JS University, Kolkata. Now, this session is handed over to Dr. Ghosh.
Okay, Dr. Ghosh, please unmute your microphone and please start the session. Yes, sir.
So should I have to use entire screen?
Yes, sir.
Okay. Let's see. Is it visible?
1 minute, sir. Yeah, I think no, it is not still visible.
Okay. Is it visible now?
1 minute, sir. No, no, sir, it is not visible still now. We can see you, but not the presentation.
No, is it okay?
No, sir, still not. Presentation is not visible.
Okay, I can. Can you just share my slides?
Yes, sir. 1 minute, sir. So please stop sharing, sir. Yeah. Yeah. Okay. 1 minute.
Yeah.
1 minute, sir.
No problem.
Okay. Now present now. New interest. Okay, 1 minute. 1 minute. Window. Yeah. Sir, I'm sharing. So kindly check whether it is visible or not.
Right.
Is it visible, sir?
Not yet.
Yeah, it is visible right now.
Not yet.
Mm. 1 minute, sir. 1 minute. Stop sharing. Your entire screen. Okay, visible in YouTube, sir. It is visible in YouTube. I think it will take some time. Okay, so okay. Just yeah, window.
Right.
Yeah, just 1 minute, sir. 1 minute.
Yeah, no problem.
Okay, so again, I'm sharing your slide, and
Okay.
okay, I think is it visible to you?
Not yet.
Okay, so 1 minute. Yeah, yes, sir. So many students saying that yeah. The it is visible in YouTube, sir. So
But I cannot see.
yeah, sir. But you can open your slide and you can start the slide from the beginning. Okay, because it is visible in the YouTube.
Okay. Let's try here.
Yes, sir.
Let me try in one way.
Yes. Everything is visible, sir. In YouTube, it is visible.
Okay. Okay. Just I'll open the YouTube then, and I accordingly proceed.
Okay.
Where is the link? Okay, here we go. Okay. But I cannot see here. Your screen is not visible over here.
Sir, now I think it is visible. Check one time. Check, please.
All right.
Is it visible,
Yeah.
sir?
Yeah. It's visible
Okay, sir.
now.
Yeah, you can start, sir.
You can just make it full screen, because what the people are writing the comments, so I cannot see. So you can just make it full screen.
Okay. Give me 1 minute,
Or
sir.
it's better. Maybe I can share that if it is visible on the YouTube, then it will be easy for me. Right?
Yes, sir, it is YouTube. Yeah, it is visible in YouTube, sir. You can start, sir.
Right. Right. You do one thing. I think it's a good idea, because if I have to see from the YouTube, I can share my I could share my slides. It's gonna be better, because people are writing comments, so I cannot able to see my slides over there.
Okay, sir. No problem. You can share, sir.
And I think, yeah, I think it will be a good idea. Shomnath, now it's visible on my screen.
Okay. Then you can share, sir. So
Okay.
then, yeah, you can share, sir. Then
Okay.
okay. Let me stop my presentation first.
Okay.
Okay,
Okay.
let me stop my presentation. Yeah, now, yeah, I've stopped my presentation. You can now present yours.
Okay. Great. Okay. Now it's visible.
1 minute, sir. It is showing it is presenting.
Okay. Let
Yeah,
me know
it
when it's
is.
fine.
Um, 1 minute, sir.
Yeah.
But in the Google Meet, it is not showing, sir. In Google Meet, it is not showing. It is not visible in Google Meet.
Just wait for a while. I think.
Yes, sir. Sure, sir. I think it is. Yeah, it is showing in YouTube, sir. You can start to
Okay.
give the lecture, sir.
Okay. Okay.
It is visible. Yeah,
Okay.
perfectly visible,
If
sir.
anything wrong, if anything wrong, you can just let me know. Okay.
Yes, sir. Sure, sir. Sure. You can start, sir.
So should I start? Okay.
Yeah.
So very good evening, everyone. Yeah. First of all, I really would like to thank Mr. Shomnath Das and Ms. Priyanka Talukdar for giving me this opportunity to present over here in a huge, great audience in this great institution. So today, I'd like to share some of my ideas and knowledges on molecular phenomena on repurposed therapeutics against the COVID-19. So this is the lecture flow that I'm going to deliver. So it consists mostly of molecular overview on the physiology and pathogenesis of the COVID-19, and why we didn't need to study the molecular view on physiology and pathogenesis on COVID-19. And finally, I will end up with repurposing of therapeutics against the COVID-19. So let's start. So in the slides, I make little bit writing because a lot of students are over here, those who have registered. So it will be easy for them to catch up what I'm trying to deliver in this lecture talk. Okay, so first, I think a lot of people have already aware of what is COVID-19. So I just make it in a very brief way. COVID-19 is an acute respiratory disease caused by the novel coronavirus SARS-CoV-2. It is also known as 2019 nCoV. COVID-19 coronavirus SARS-2 belongs to the beta coronavirus genus It's basically originating from the bat. So it belongs to beta coronavirus that can infect the mammals are the zoonotic pathogens. That means they can actually carry out their life cycles into different animals. It could be in a bat, it could be in a human, because there is some common receptors on their surface where they're going to attach in the host and can cause severe respiratory disease in human. I will show you a nice pictorial presentation in the next slides. Unlike other viruses in this family are the SARS coronavirus, MERS coronavirus. It shows that the COVID-19 SARS-CoV-2 has approximately 79% sequence similarity, identity to the native coronavirus. Also, the SARS-CoV-2 has also resembles 50% as per the sequence identity with the MERS one, which is another family or belongs to the beta coronaviruses. Now, this is a nice pictorial presentation, which it is being published by the Tang group, and it shows like how the novel coronavirus has derived from the very ancient one. So, as we know, because we are not going to discuss about the taxonomy and how they derive by the speciation, but it's like a nice way how it's presented over here, like how the novel coronavirus has been developed. So you can see it's a nice structures, different kind of surface proteins are present, mostly membrane glycoproteins, spike proteins, nucleocapsid proteins, the RNA genomic material and envelopes. So these are the basic similarities that there in the native one, and due to speciation, due to adaptive evolution in the nature, they have changes their sequence pattern. So mostly the sequence motifs or the domains has been gradually being changed due to the adaptive pressure which is coming from the environment. And actually then it's coming and come up to a novel beta coronavirus in this family. Now, in addition, the homology modeling, it's a molecular docking process, which shows the COVID-19 SARS-CoV-2 has a similar receptor binding domain structure as SARS-CoV, which is a primitive SARS-CoV or coronavirus, which suggests the SARS-CoV-2 uses the similar kind of receptors, that is ACE2, which are present on the human respiratory cell surfaces, and which actually induces the infections when the spike protein and the ACE2 receptor interacts with each other. So this is the basic molecular structure, most on the surface topology. The structure of COVID-19 mostly consists of the spike protein, which shows S, the hemagglutinin esterase dimers that represented as HE protein, a membrane glycoprotein M, an envelope protein E, and nucleocapsid protein N, and RNA as a genetic material. And it has also specific enzyme that is RNA-dependent RNA polymerase, that is reverse transcriptase. And all these variations due to the environmental stresses mostly governed by this highly error-prone reverse transcriptase enzyme, which encoding different proteins and doing lots of misbase incorporation and mutations, which are present on the surfaces of the novel coronavirus. So let's discuss a little bit about the different surface topology corresponding proteins, which actually most interesting and important for repurposing or development of the drug or different kind of vaccines in current scenario. So spike protein is heavily glycosylated, which utilizes N-terminal signal sequence to gain the access of endoplasmic reticulum and mediate attachment to the host receptor. It is the largest structure and make distinct spike because they have a very distinct motif of amino acid residues and moieties, which are present in the primary or secondary structure on the surface of that virus. And SARS-CoV-2 is S protein is cleaved by the cellular furin-like proteases, which is TMPRESS2, which is very important. It plays very important role for the maturation of the spike protein SP6 or SP4 into two separate polypeptides are S1 and S2. S1 also consists of receptor binding domain, that is RBD, receptor AC2. The nucleocapsid protein, termed as N, binds to the RNA in vitro and heavily phosphorylated, because as we know, the phosphorylation and dephosphorylation plays a very important role. As we know, in karyotic system, we have the nucleosomal structure, and a lot of acetylation and phosphorylation, dephosphorylation phenomena actually accelerating or deaccelerating the gene expression pattern. So similar way, nucleocapsid protein also plays a very important role for winding and rewinding of the RNA, which is the genome. And when it's highly phosphorylated, it somehow get unwinded. So N protein binds to the viral genome in a binds on the string type configuration. The protein likely helps catheter the viral genome to replicase transcriptase complex, that is the RTC, and subsequently package the encapsulated genome into the viral particle. Similar way, envelope protein E is also found in very small quantity, in a minute quantity within the virus particle. It is most likely a transmembrane protein because they have a transmembrane helix. It's similar like the 7-helix receptor structure like eukaryotic system, but in a very simpler way and in very small molecular weight inside. With the ion channel activity, the protein facilitated the assembly and the release of the virion particle. And other functions such as it also exchanges the different ions is very important for the osmotic balance when they are get entered into and multiply inside the host. It is also necessary to it is also very necessary for the viral replication and also for the pathogenesis. So this is a very nice pictorial presentation which is being published by the Wellingbury, and it published in Science of Total Environment in 2020. He's a very great friend of mine, and he did work very nice on COVID-19 and specifically with the pathogenesis of this particular virion particle. So now let's discuss about what this picture or pictorial presentation depicts. So number one, if you follow the picture, you can easily understand what I'm delivering now. So it has been confirmed that the SARS-CoV-2 uses the same cellular entry that, as I mentioned earlier, it has a ACE2 receptor, and this ACE2 receptor get activated by proteolytic activity of the TMPRESS2. Now, when this ACE2 receptor get activated, and then the SARS-CoV surface receptor, spike 4 or spike 7, 6 protein, they get started interacting with each other. So this is the first point of entry through which SARS-CoV-2 get invaginated inside the suitable host, which having ACE2 receptor on their surface. The human ACE2, then it's found in the lower respiratory tract of the human. That means the ACE2 receptor, they mostly embedded on the surface of the lower respiration tract. Works like a cell receptor for the SARS-CoV or CoV-2 that regulates both the cross-species and human-to-human transmission. Because I'm not going to go into the detail like what are the mode of transmission, mostly like droplet or the aerosol borne, all those things, because everything when it gets started, get stuck into it, and then how they get inside the host and do the replication and then come up with the complete virion particle. Now, the virion is glycoprotein present on the surface of the coronavirus that attaches to the receptor ACE2 on the surface of the human cells. It has been also shown that the S protein, the spike protein, and the ACE2 binding efficacy is 10 to 20-fold higher than that of the SARS-CoV. That means the novel coronavirus having 10 to 20% higher affinity, avidity, and interactability with the ACE2 than the native or the primitive SARS coronavirus. In the case of SARS-CoV-2, the cleavage of the trimer S protein, the spike protein, is initiated by the cell surface associated transmembrane protease serine 2, that is TMPRESS2. And it also associated with the cathepsin protein, which also helps to accelerate and maturation of the TMPRESS2 protease, which is serine-dependent protease molecule. At the same time, the possible molecules facilitated the membrane invagination for the SARS-CoV-2 endocytosis are not really being very still clear how it getting endocytosized inside a vesicular form. However, it has been observed that the SARS-CoV-2 may readily transmit the widely causing less severe human infection rather than the human SARS, the primitive coronavirus. The S-glycosylation includes the two subunits, as I told earlier, that is S1 subunit and S2 fragment. The S1 is responsible for the determination of the viral location of the protein, facilitate or accelerating the viral cell membrane fusion by two tandem domains, which are very conserved for the novel coronavirus. It is mostly the haptid repeats, HR1, and the haptid repeats 2, HR2. These are the specific motifs. The genomic RNA of the novel coronavirus of approximately 30,000 nucleotide encodes structural proteins and also non-structural protein residues of the virion particle that have a critical role in the viral RNA synthesis called the replicase transcriptase protein. At the least, on each specific protein, the non-structural protein NSP2 and one structural protein, the nucleocapsid protein N, involved in the viral RNA synthesis. It's accelerating the RNA copy number increase inside the host when it gets endocytosized. The expression of the coronavirus replicase transcriptase protein complex, which is associated with some gene encoding functions, is mediated by the translation of the genomic RNA. That means the RNA will it's converted into the complementary follow the corresponding different kinds of protein molecules which are being recognized and is useful for the virion to make the complete virion particle. The replicase transcriptase proteins are encoded in an open reading frame. As you know, open reading frames are the specific sequence which starts from the start codon and terminates with the stop codon, and it encodes specific polypeptide residues. Then it gets folded and become biologically active functional enzymes of catalytic components. So here, these are mostly two important ORFs are there, ORF1A and ORF1B, and are synthesized initially as two large polypeptides. One is PP1A and PP1B. You can easily see here PP1A and PP1AB. The synthesis of the PP1AB involves the programmed ribosomal frame shifting during the translation of the ORF1A. During or after the synthesis of the RNA, the genetic material, these polypeptides are cleaved by the viral encode. That is the post-translational modification. Unless they cleaved, their signal peptide gone, the specific signal on the N-terminal end, it's gone, they cannot able to become biologically active and do their proper functioning, which is responsible or accelerating the virion particle budding outside the host when the titer has been reached. So these are mostly papain-like, PL-Pro, and the chymotrypsin-like folds into the 16 protein residues are there. Now, the NSP1 and NSP11 are encoded in the ORF1A, and NSP12 to NSP16 are encoded by the ORF1B, because these ORFs are the overlapping ORFs. So it has a lot of ATG and also TAA in between the entire this entire open reading frame. That means they're differentially expressing depending on which protein they require for their functionality. The replicase transcriptase protein, together with the other viral proteins and possibly cellular proteins, assemble into membrane-bound replication transcription complexes, which is so-called the RTC complex. These complexes accumulates at the perinuclear regions, are encoded with the double membrane vesicles. It's very important because they require the complex to be assembled and get properly maturated along with the help of different accessory proteins. Some accessory proteins are being provided by the host itself when the virulent particle actually hijack the host defense mechanism. So hydrophobic transmembrane domains are present NSP3, NSP4, and NSP6 likely serve to anchor the nascent PP1A and PP1AB polypeptide proteins to membranes during the first step of the RTC formation. Finally, the virion-containing vesicles fuse with the transmembrane of the cells to release the complete assembled active virion particle. The virus then attaches in a new cells in their adjacent cells which having a C2 receptor, and then the cycle goes on and is being repeated. So this is the exact molecular mechanism which is being understood by different proteomics, transcriptomics, and genomics analysis, by comparative genomics and proteomics. And that's the thing related with the pathogenesis of the novel coronavirus till that. So in a very simplistic way, I'd like to try how this interaction is being going on. You can see the SARS-CoV-2, which having specific spike protein on the surface. It binds with the ACE2 receptor, which is present in the lower respiratory tract of the lungs. Then it accelerates the viral entry, the host cells, fusion, and viral replication. So it can go into the two directions. Either it can show the cytotoxic effect and alter the lungs pathology, and it worsens the lungs damage and causes the acute respiratory distress syndrome, or CRC, the Ross formation, and shock on the day. Now, on the other way, the SARS-CoV-2 directly inhibits certain immune activities, like it somehow inhibits the innate immune response, interferon pathways. It increases the viral load and actually, which induces the infection. When the titer getting more and more, then eventually it will just burst out the cell and make a lot of mucus and membrane that can cover the oxygen mass transfer in the lungs. So that's the causes the acute respiratory syndrome or the failure. The similar way, it also including the failure in the adaptive T cell mediated damages. So now, as I mentioned in the previous slide, this is causes the cytokine storm, which is mediated by the interleukin-6 and others. I think the next speaker will elaborately explain about how this cytokine storm has been occurring, but it's in a very precise way. Hello, am I audible? Hello.
Yes, sir. Yes, sir. You are audible, sir. You are audible, sir.
how the cytokine storm has been induced by the SARS-CoV-2 novel coronavirus. So you can see the SARS-CoV-2 infection first initiated by the S1S2 cleaved fragment. It interacts with the ACE2, which is being activated by the TMPS2, and that induces the necrotic factor kappa B, and which actually induces the interleukin 6, followed by the AMP and cyclic AMP cyclic formation. And this interleukin 6 gets activated, and it actually causes, when the interleukin 6 titer is quite high, it makes a storm-like situation inside the host cells, and it induces the inflammatory cytokine that is called ARDS. Am I audible? Hello? Hello? Am I audible?
Yes, sir. You're audible, sir. You're audible, sir.
Okay. 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 Now, next, then what should be the take-home message from this pathogenesis and the physiology? Why physiology and pathogenesis is very important, and what will be the take-home message? What you are going to what you did understand from this presentation? So, in a very nice pictorial way, which is published by the Bhupati, is a it seems like he's an Indian scientist, but he's an Indian, but he's working in the United States, and he published a very nice pictorial presentation of how the pathogenesis is being carried out. So if you can make them different steps and the segmented form, then you can say the first step is the SARS-CoV-2 entry point. Second one is the membrane fusion. Third one is the RNA release, and then followed by the translation process and post-translational modifications. And then RNA replication will begins, and then it will go for the translational steps, and finally the RNA will be get packaged, and then the complete release of the virion particle. So this is the very nice and very systematic presentation in a very brief way how the COVID-19 carry out the pathogenesis in their suitable host, where they have the ACE2 surface receptor, which is being formulated by the activity of TMPS2. So is it clear till now that what I have explained? Hello?
Yes, sir. Yes, sir. Yes, sir.
Okay, so should I move forward for the next phase?
Yes,
Okay.
sir. Sure.
Okay, thank
Yes,
you. Okay, now the question comes, why do we need to study this molecular view or molecular phenomena get understood by us based on the physiology and pathogenesis of the COVID-19? That means the question itself says, answering like how this physiology and pathogenesis phenomena associated with the COVID-19 accelerating the repurposing uses of the different kind of drugs and the vaccines. So these are the basic answers. This question actually answers it mostly useful for the target selection of different therapeutics. It could be repurposed therapeutics, it could be newly developed therapeutics, it could be newly developed vaccine or repurposed vaccine, whatever it is, mostly used for preventing or for destroying the COVID-19 pathogenesis and protect our lungs from get infected and make it the situation more worse. So the first target selection important for the therapeutics one, that is the application of the repurposed drugs, novel drug development, and the development of the different vaccines. So now if you know the surface topology or the pattern of different proteins which are protruded outside the novel coronavirus, and if you know which are the different steps which are involved during the pathogenesis, and what are the different components and modules which are responsible for the different steps of the pathogenesis, then it will actually accelerating the science or accelerating the researcher's mind to develop new repurposed drugs, or you can develop new drugs, you can develop new vaccines as well. So now next, move on to the next phase of this presentation. So why repurposing and development of therapeutics against the COVID-19 is very important. So I actually try to divide this into two different segments, one for the candidate drugs, mostly therapeutics like antiviral compounds, different kind of anti-malarial drugs, different antiviral drugs, different bacterial drugs that can use for the checkpoint or for the arrest of different other viruses like MERS, SARS, Ebola, Zika virus. These are belongs to the similar kind of family. So they should have been working against the novel coronavirus. On the other hand, I segmented into the vaccination strategy. So mostly related with the candidate, which are going to be the potential vaccine after having the preclinical steps approval. So you can say here, this is very important as I discussed in the previous few consecutive slides. These are the different surface proteins. These proteins are actually interacting with this one. That means this is the first checkpoint, that is the entry point. If you can find out some specific drug molecules, drug candidates, which can interact with this kind of proteins, then easily it can hinder this ACE2 receptor interaction. So this is the first mode of entry will be inhibition if you use or repurpose some drug which can interact with those kind of molecules. Similar way, the next steps is the proteolysis. Now, if you find some kind of drugs or some other molecules, bioactive molecules, which can interact with these different protease molecules, then the proteolysis may not be taken place. And if there is no proteolysis, then the consecutive replication or transcription complex will not be formed. So now these are the different checkpoints which are actually being determined by if you have a complete study about the pathogenesis of this process. Similar way, if you consider the vaccination strategy, you can find different vaccine candidates. They can be applied for the protein subunits. Like if you consider the S protein, E protein, M protein, these are the different vaccine candidates which can be applied for the protein subunits.
Hello.
Viral peptides, the VLPs, you can also develop. Inactivated vaccine can also be tried. Different procedures are there, but I'm not going to go into the detail on those things. And then mRNA vaccine can be developed in that particular checkpoint if the reverse transcriptase works. And the DNA vaccine can also be developed. And replicating and non-replicating viral vector can also be designed and tried out to work against the COVID-19 as a potential vaccine. Similar way, live attenuated vaccine is as an example. Okay. So you can see different drugs have a different targets. Like if you consider here, this is the difference in this particular drug is actually works interacts with the spike S protein. Now when it interacts with the spike, it inhibits the initial entry point interaction with the AC2. That means the mode of entry will be somehow hindered. The similar way, if you find some specific like ritonavir, leponavir, these are the different kinds of drugs which are actually protease inhibitors. So mostly SGLT2 pro inhibitors or the CCL pro inhibitors, also RDPP, that is reverse transcriptase enzyme inhibitors. So if you use these particular drugs, because these drugs are not naturally used for the SARS novel coronavirus, it is used for the HIV treatment. Now, people try to repurpose it. The basic meaning of repurpose means the antiviral compound or any bioactive compound which is used for some native virus, which are already existing. Now, if you want to use that particular virus for a new viral infection prevention, then it's called repurposing. You are using for the different other purposes. But you have to make sure, like, is there any similarity with the reverse transcriptase, which is present in the SMARS? It is also similar like in the SARS-CoV-2. That you have to go for comparative analysis, comparative sequence analysis, motive analysis. That actually gives you a clear idea. That's why physiology and pathogenesis plays very important role to understand which drug and when it will be applied and which checkpoint will be met by if you use that particular kind of antiviral compound.
হঠাৎ করে কি হলো আবার?
হ্যালো, শোনা যাচ্ছে?
এক মিনিট, হ্যাঁ আমি তো শুনতে পাচ্ছি স্যার, একবার জিজ্ঞেস করে নিচ্ছি।
Right.
এক মিনিট।
আসলে নেটটা এত উইক না, সেই কারণে সমস্যা।
নেটেরই প্রবলেম হচ্ছে।
Hello? Yeah, I think it's audible, right? Okay. Should I start?
Yes, yeah. You said.
Okay,
Yeah.
so
Trust.
in these slides, I try to mention like how you can choose the target based on the different kind of checkpoints. So as I mentioned earlier, so now there are few mechanisms which are known, like when you're using chloroquine or hydroxychloroquine, how it works. The basic mechanism action of this anti-malarial drug would result in an increase in the endosomal pH, as I said earlier, by preventing the fusion between the virus and the cells, or it interfering in the glycosylation process of the cellular receptor of the coronavirus. So in addition, it is hypothesized also that these antimalarial have an immunomodulating effects that contribute to the increasing their efficacy in vivo. But it's in vivo, but in vitro, nobody has actually validated this kind of hypothesis that has been deduced over here. The similar way, as I said, remdesivir is another very good antiviral compound against the HIV, but it repurposed for the COVID-19. The probable molecular mechanism of the remdesivir against the SARS-CoV-2, number one, the schematic diagram shows the SARS-CoV-2 viral entry and its RNA synthesis, which can be blocked by the remdesivir. So you can see remdesivir is actually making the RDBTP complex, and that interfere with the RNA-dependent RNA polymerase activity. Then the viral particle and their corresponding RNA not get it replicated due to the deficiency of the ATP and their corresponding complex formation. That finally it ends up with the inhibition in the viral RNA synthesis and increase in the copy number. That means there are no further budding and no fusion and no release of the variant particles. So it's like that. Now, it is the final one that has been science paper. It's being published by the Cohen Research Group. So it's very interesting and very impressive. This is very authentic paper, and it shows the basic mechanism. So if you have a time, you can read this particular science article, which is being published by the Cohen Research Group, and you will come to know how molecular interaction is being going on, because we have a time constraint, so we need to be limited towards specific lectures on that. So you can see the monoclonal antibody, which is conserved in the plasma that interacting with the spike protein. The similar way, the chemostat mesolytes also interacting with the TMPRESS2 activity. Similar way, the chloroquine, hydroxychloroquine that can inhibit the endocytosis process. The ritonavir and the leponavir actually inhibits the proteolysis process. Also, remdesivir interacting with the RNA-dependent RNA polymerase activity, and ultimately and overall, the variant particle release and budding will not be taking place. That means viral infection or pathogenesis will be somehow getting inhibited over here. Now, this very interesting paper recently come out in the Cell paper by a Capitals research group. It shows that the plant biotechnologies can also participate and involved in this particular process. So you can see over here, mostly tobacco, this is called the molecular farming, how you can use the plant-based physiological approach to develop different therapeutic antibodies, different therapeutic bioactive molecules with the involvement of the antibody generation and immunization process. I'm not an immunologist, so I have not a complete idea how this immune responses is being going on over here, but this is a nice presentation in this particular paper. So you have a time, you can read how tobacco and the maize plant can be useful tool for large-scale production of the SARS-CoV-2 surface-specific proteins and their corresponding antibody that can be used as a therapeutic compound, and it can also be used as a probable vaccines. Now, Dr. Bhomik will continue on that particular section, which is vaccination. So I'd like to restrict over here myself, but in a brief way, maybe it will help Dr. Bhomik to go for the further detail on that particular presentation that will be followed after me. So mostly the therapeutic purposes, different viruses are used and different approaches. So you can see as a vaccination, definitely. So mostly viral vector-based, inactivated viruses, subunit viral proteins, viral-like proteins, DNA vaccines, and attenuated virus. These are the different strategies. So this is a nice pictorial presentation, but I'm not going to go into the detail because Dr. Bhumik will continue and further go for the analysis and elaborative discussion on that. So this is a very nice way to depict how the successful vaccine can be developed against the SARS-CoV-2 by involvement of different kind of immunological activity inside a suitable host, by clearance of the viral infection and also enhancement of the viral infection consecutively. So now we need to know why COVID-19 vaccine is very important, because usually, if you can see the classical vaccine developments, the preclinical stage takes 30 months maximum. All the things, all the phases taking lot of time, but we really don't have that much time because the outbreak is very panicking and very devastating. So everything is goes down to months to days. And you can see in the preclinical stage for the classical vaccine, it requires 30 months. In clinical, the 0 months. So we really don't have enough time to develop anything else. Even for the phase 3, if you consider phase 2, it's look at 32 months. For in case of COVID-19, it's only 6 months there for accumulating hundreds of volunteers to apply. And it's really very quicker because approval, manufacturing, vaccination due to the demand, public demand, and it's really health issue. So billions of doses an individual just need to be completed within 6 months, which is usually required by the conventional approaches for 12 to 24 months. It's a huge pressure on the researchers. So is common people, layman always saying, scientists, what they're doing? Really, they're doing very hard work and the doctors, the nurses, all the people, they are highly indulged to help all the common people. So we need to also cooperate with them and make proper safety and prevention measures that has been broadcasted by the television and said by the eminent doctors. So what are the take-home message then? So this is actually the take-home message, nice presentation by this research group, which is published in April 2020. So you can see it's a nice way. So you can take home these particular messages as a note. So target cellular signaling pathway, these are the target, blocking viral entry point, interfering with the endocytic pathway, like chloroquine, hydroxychloroquine plays. So we need to find some other bioactive compound that can place far better, because chloroquine, hydroxychloroquine has a lot of side effect in the dilation of the blood, RBC, lysis, all those things. So we need to find some bioactive compound which is better substitute than chloroquine or hydroxychloroquine. Blocking the polypeptide, the polyprotein translation and processing S1, S2, PLOs proteins, oligonucleotide gene therapies, the RNA-based therapies, supplement of the nutraceuticals, some immunoboosters that can be found out by the natural medicinal plants of India. But that has to be properly characterized, not just like a story, take the entire extract, put in the tea, and not like that. So you have to proper characterization by the FTIR or by the mass spectroscopy. The modulation of the immune defense mechanism, vaccine development, various antiviral mechanism has to be deduced. It's very important. We need to find out how antiviral compounds specifically targeting what particular segment of the pathogenesis cycle. And finally, targeting the viral RNA-dependent RNA polymerase. Because even if you develop one vaccine, that may not be persist for the next year, because the reverse transcriptase, which is the RNA-dependent RNA polymerase present in the SARS-CoV-2, it has high error rate during the polymerization. That means spike protein 7, which is, if you develop some vaccine or some anti-therapeutic compound, again, SP7, maybe the next year, SP7 get mutated. So maybe the existing vaccine may not be properly functioning in the next year. So that study has to be done very carefully and find out how least efficiently RNA-dependent reverse transcriptase working and how we can repair this particular reverse transcriptase enzyme that can causes least amount of mutations. So this is another very interesting, I find just before ending up my presentation, Rahman is published these particular things. You can see it's a nice way out. It's not related directly with the biology, but polymer chemists, also the textile engineers can get involved. So what they did, they take the Indian medicinal plant, the extracts, having different anti-inflammatory immunoboosting ability. They blend it with the cotton fibers and then go for the electrospinning process, developing nanofibers. And then they collect these particular nanofibers, which are treated with these Indian medicinal extracts, and then incorporate into the mask. So that means this is a bioactive mask having different filtration unit that protect the viral get enter into our nasal route. That means they cannot get inside the lungs and not getting chance to react with the AC2 receptor. So these are the things that we are currently working in the microbial engineering and algal biotechnology lab. one of my students is working on finding different novel targets. These are the very novel targets. You can also have a look on that particular paper from Land Cancer, published from UK, a very authentic research journal, and this article. So it shows a lot of different other target molecules can be targeted and repurposed for some other drugs in future. So based on this particular literature, because literature review is very important, so and based on this particular target selection, we are doing the different kind of studies, mostly the screening and identification of novel therapeutics through the repurposing approaches. These are different computational biological approaches, bioinformatics approaches to develop against the novel targets of the COVID-19. So I will wrap up my presentation over here. Smaller compared to the house. A lock is much smaller compared to the door. The key is the smallest of all above. But a key can open entire house. Thus, a small, thoughtful solution or idea can solve the major problem. That means corona is a major problem. Only we need to have a very honest, very innovative idea. It may come from any mind or any brain or any cerebral hemisphere, but it has to be worked out. This idea should be very useful, and that can just work out bingo to develop a new therapeutic approach that can completely eradicate the era of coronavirus COVID-19. Thank you very much for your attention.
Thank you. Thank you so much, sir, for the nice presentation. There are so many questions from the audience. So can I start the question answer session, sir?
Yeah, audience, I think audience also know when you introducing me, I'm not really a virologist. So
No,
a biology
still,
and
sir.
bioinformatics person. Okay,
Yes,
go
sir.
ahead.
Okay, so the first question is, can RNA interference-based therapy be useful in COVID-19 treatment?
RNA interference therapy can also be possible, because as we know, we have also developed one kit, which is called Feluda kit, mostly based on the CRISPR-Cas. So
Yes, sir.
CRISPR-Cas is nothing but the daughter approach, which actually resembles with the RNA I complex formation. It has a PAM sequence, it has a specific 5-terminal ends homologous sequences which can interact with RNAi can be used, can be applicable, but it still depends on what are the targets and how you are going to design the target based on the PAM sequences.
Okay, sir. Okay, sir. Thank you, sir. Next question is, Shreya says that recently I heard a news that a person who recovered from COVID-19 is again infected. So how is it possible, sir? I mean, once the memory cells are formed, the body usually detects the pathogen. Then why second time infection is occurring?
As I said, maybe the virus is it's highly mutable. The B cells or the memory cells, which has been developed by the B cell proliferation or antigen presentation by the class 1, class 2 MHC or the APC, CD4, CD8 interaction, all are based on the hapten. Now, if your hapten is SP6 or SP7, and now after 6 months, the motifs in that particular polypeptide, amino acid residues in the motif has been changed due to the errorness of the reverse transcriptase. That means your memory cell may not be able to recognize SP6, which was before SP6. Maybe now it's SP6 become mutated.
Okay.
That means the recognition pattern may be changed. Host may not be recognized this SP7, which has been made earlier.
Okay. Okay, sir. Thank you, sir. Next question is, what do you have to say about the production of bioengineered free ACE-like receptors? Do you have any update on that, sir?
Can you just repeat the question?
Yeah, sir. What do you have to say about the production of bioengineered free ACE receptors, ACE-like receptors? Bioengineered?
What we are going to do with this engineered AC2?
Yeah, Ankit asked this question.
If I will really understand what he meant, he told, like, if he engineered the ACE2 and then replace the wild or the primitive one, does it solve a problem but mode of entry? But the question comes, how it will be just presented on the surface of the lower respiratory tract, on the tracheal cells, because it's a native process. Because if you develop or inject some ACE2 receptor which is mutated, it has to be protruded outside. And what will
Yes,
be
sir.
the driving force? What are the channel proteins which actually make that protein to get exposed and get transmembrane locked on the outer surface? Because it has to be placed in a certain place, right?
Yeah,
On
yeah.
the membrane. Now, who will actually accelerate this process? That has to be first resolved. AC2 transporters. Ankit, you should first try to understand what are the channel or the transporter proteins. Which proteins actually helps to trafficking of ACE2. If you find out that particular kind of trafficking protein which are correspond to the ACE2 receptor, then only it will be fruitful or feasible technology to develop engineered ACE2 by altering their specific protein motifs which interacts with the SP spike proteins or the envelope proteins.
Yes, sir. Okay, sir.
My answer
Yeah.
is you have to find out certain protein which actually helps to traffic that ACE2 and then just embedded on the membrane surface. That is very important. You need to understand the interaction. Which protein is actually acts as a carrier for the ACE2 to get exposed or protruded outside or presented on the tracheal cell surface, right?
Yes, sir.
Right.
Okay. Okay, sir. Next question is from Shomo. He says that is there any relation between the immunity of a person and the person being asymptomatic to COVID-19?
Hello, can you repeat, Somnath?
Yeah, yeah, yeah. Is there any relation between the immunity of a person and the person being asymptomatic to COVID-19?
I don't get the question. Can you repeat?
Yeah. Is there any relationship between the immunity of a person and the person being asymptomatic to COVID-19?
If I correctly understood, the question is one person is get infected, another person is infected but not showing any symptom. Is there
Yes,
any
yes, yes.
difference? Is there any difference
Yeah.
with? Is there any
Yes, sir.
difference?
Yeah. Is there any
Definitely,
difference
because
and is there any relationship between these two?
relationship. What kind of relationship? Because one is showing immune response, another one is not showing immune response, right?
Hmm.
So this is the first line of entry because the virus particle is there in the trachea. Maybe they cannot able to replicate. First
Yes,
line
sir.
of response will be definitely there. It's asymptomatic, but maybe some kind of minor alteration in the body will definitely take place because when some hapten or foreign particle get intrude inside, definitely there will be some kind of alteration. Maybe the alteration level or efficacy is so low it's not in the detectable level, but definitely certain B cell proliferation definitely taking place and the antibody generation in a very minute titer definitely there in that particular host who is an asymptomatic carrier.
Okay. Okay, sir.
Right.
Next, Vishal says that ACE2 receptors in eyes are also sensitive for infection with COVID-19. So,
Yeah, because
yeah.
yeah. Please go ahead.
No, sir. This is the question. So, I mean, he says that ACE2 receptor is also, through which the infection can be there through the
That
ACE2 receptor. Yeah.
it is not only on the nose, not only in the eyes, it is also in the gastrointestinal tract. So you can also
Okay.
get infected like similar way like entamoeba histolytica. So if you are get infected with some kind of feces by some infected people, you can get infected. But the question comes,
Hmm.
how efficient it will be, how efficient will be the mode of entry, how efficient will be the checkpoint overcome by the virus. That's very important. That's why if you just forget about the intestinal tract, if you consider the eyes, Vishal's question, that's why
Yeah.
doctors saying when you are using your bare hand always sanitize after 1 or 2 hours, because if you somehow rub your eyes you may get infected. There is a possibility is always there because it's a probability. Definitely you have 50-50 chance. If
Yes,
you
sir.
are immunocompromised, maybe that time at that particular stage, maybe you get infected if you rub your eyes with your infected fingers. It could be.
Okay, sir. Okay, sir. Next, Chondima asks that, sir, is okay, who is already COVID positive for once, but after recovery, is or she has any chance to be COVID positive again?
The previous question answer her question,
Yeah,
right?
yeah, 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 yeah. Okay.
He or she can reinfect it again.
Yes, sir. Okay, sir. There are so many questions are there, so I'm just finding the questions. Um, okay, sir. Can
Doctor,
plasma therapy? Yeah,
yeah.
can plasma therapy will be effective?
Haha, I
Because
think Dr.
yeah,
Bhumi can give better answer than me whether plasma
okay,
therapy will be working or not. I think it's better to ask him. He's the right person.
okay, sir. Um, okay, and 1 minute, sir, if possible. Okay, sir. Okay, again, the question comes from the vaccine part. Okay, I think it will be answered
Yeah,
by Dr. Bhumik. I
Bhumi,
think.
I think it's better. He's waiting for long. I think he should
Yeah.
move forward. Yeah. Okay.
Okay, sir. Okay, sir. Thank you so much, sir, for the nice
Welcome.
presentation.
I'm
And
I'm
we are
I'm
really
I'm
happy, sir.
grateful. I'm obliged. Thank you. I appreciate it. Thank you.
Okay, sir. Thank you, sir. Okay, so thank you, Dr. Ghosh. So now we are going to our next speaker, Dr. Bhumik, and the session will be handed over to Shomo Sharati Ganguly. So over to Shomo. Shomo, are you there?
Yes, good evening to all of you. Our next speaker is Dr. Subhrajyoti Bhomik, who is the uh, clinical director of academics, research and quality in Peerless Hospital, Kolkata. He is certified in patient safety from Johns Hopkins University, USA. Uh, he was the winner of the ISQ UA LEAP Patient Safety Fellowship Award in 2019. He is the NABH assessor for hospitals and ethics committee. He was the honorary lecturer of Institute of Chemical Sciences, University of Birmingham, UK. He was formerly attached to Stanford University School of Medicine, USA. Over to you, Dr. Bhumi. You can start the session, sir.
Yes, sir. Yes, sir. You're
Yes, sir.
audible, sir.
You're
Yes, sir. It is visible, sir. Sir, sir, sorry, sir. Yeah, you are not audible, sir. Just 1 minute, sir. I think there is some problem. 1 minute, sir. Hello. Yeah.
no, sir. You are not audible in YouTube. Sir,
স্যার আরেকবার একটু বলেন না না এখন অডিবল হয়নি স্যার 1 মিনিট 1 মিনিট স্যার ওকে স্যার স্যার আপনার আপনার মাইকটা অন আছে তো আর আর হচ্ছে আপনার সিস্টেমের মাইক সিস্টেমের সাউন্ড অন আছে ওকে 1 মিনিট স্যার দেখছি 1 মিনিট স্যার I can, sir, I can hear you in Google Meet, sir, but it is not coming in YouTube, so that is why we are trying to fix it, sir. 1 minute, 1 minute, sir. হ্যাঁ, সৌম্য একবার একটু সাউন্ড দেখ, একবার চেক করতে ওদিক থেকে কোনো প্রবলেম হচ্ছে নাকি। এখানে তো কোনো প্রবলেম হচ্ছে না, ঠিকই আছে সিস্টেমে।
না স্যারের সাউন্ডটা গুগল মিটে শোনা যাচ্ছে, বাট ইউটিউবে এখনো শোনা যাচ্ছে না।
সৌম্যরটা ইউটিউবে শোনা যাচ্ছে। আচ্ছা আচ্ছা, হ্যাঁ হ্যাঁ হ্যাঁ। সৌম্য একবার একটু সাউন্ড দেখ, একবার চেক করতে ওদিক থেকে কোনো প্রবলেম হচ্ছে। এখানে তো কোনো প্রবলেম হচ্ছে না, ঠিকই আছে সিস্টেমে।
স্যারের সাউন্ডটা গুগল মিটে শোনা যাচ্ছে, বাট ইউটিউবে এখনো শোনা যাচ্ছে না। আমার সাউন্ডটাও ইউটিউবে শোনা যাচ্ছে, একটু ল্যাগ আছে না ইউটিউবে।
সৌম্যরটা ইউটিউবে শোনা যাচ্ছে। আচ্ছা আচ্ছা, হ্যাঁ। ওকে স্যার, 1 মিনিট। হ্যাঁ, না স্যার আসছে না, আওয়াজ আসছে না, কেন আসছে না? 1 মিনিট। সৌম্য, তোর ওখান থেকে তুই একটু, সবাই একটু সাউন্ডটাকে অফ করি, ঠিক আছে? সবাই সাউন্ড অফ করি, স্যার একবার বলুন, দিয়ে তারপর চেক করছি যে শোনা যাচ্ছে কিনা। আর না হলে আমি একবার এখানে বলে দেখছি যে সবাই শুনতে পাচ্ছি কিনা। ইজ ইট অডিবল নাও? 1 মিনিট স্যার। ইয়েস স্যার, 1 মিনিট। অডিবল? Hello everyone, can you hear me? Hello, please write yes if I'm audible. ওখানে কি শোনা যাচ্ছে? আসছে না? Yes, I'm audible. Okay, আমি তো অডিবল। দেন তাহলে স্যারও এবার আই থিঙ্ক বলতে পারবেন। হ্যাঁ স্যার, ইউ ক্যান কন্টিনিউ। আই থিঙ্ক we are audible, স্যার। Yes স্যার। ওকে, 1 মিনিট। স্যার আপনি একবার একটু স্টার্ট করেন, ওকে? দিয়ে আমাকে তো সবাই শুনতে পাচ্ছে ইউটিউবে, ওকে? আর আপনি একবার স্টার্ট করেন, দিয়ে আমি এখানে আস্ক করে নিচ্ছি যে শোনা যাচ্ছে কিনা, ওকে? স্যার, আমি জিজ্ঞেস করছি। ওকে স্যার, আমি জিজ্ঞেস করলাম অল দ্য অডিয়েন্স, ওকে? 1 মিনিট স্যার। না স্যার আপনাকে শোনা যাচ্ছে না। ইয়েস স্যার, আই ক্যান হিয়ার স্যার। স্যার আপনি একবার এটাকে মাইকটাকে অফ করে আরেকবার অন করেন তো স্যার, আই থিঙ্ক। কারণ হ্যাঁ, আমাদের সবাইকে শুনতে পাচ্ছে, ওখানে একবার মাইকটা এটা করে দেখেন। হ্যাঁ Yes, sir. Yes, sir. Yes, sir. Sir. I can hear you. I'm asking. Asking everyone. Okay. Yeah, yeah, yeah. এক মিনিট স্যার আসছে না ওখানে এক মিনিট স্যার এক মিনিট আমি আজ করছি দীপ কি শোনা যাচ্ছে কি ওদিকে স্যারের আওয়াজটা স্যারের আওয়াজ শোনা যাচ্ছে না কেন স্যার যখন স্টার্ট করেছিলেন তখন কি শোনা যাচ্ছিল
আমার মনে হয় আপনি একবার হ্যাঁ আপনি একবার লগ আউট করে দেখুন আমি লগ আউট করেছিলাম তখন কাজ হয়েছে একবার করে দেখুন মনে হয় কাজ হবে আচ্ছা
hello am I audible to all please write yes if I'm audible hello Okay. Yes, so there is some, there is some technical problem. Okay, so just give us 5 minutes. Dr. Bhumik will be there in next 5 minutes. Okay, so before that, Dr. Bhumik is present. Yeah. Yes. So 1 minute, I'm asking everyone. Okay. Yes, sir. Okay, 1 minute, sir. আচ্ছা, এবারে কি শোনা যাচ্ছে? স্যার 1 minute, স্যার। আমরা within 5 minutes, we are just restoring our system. Okay, so I would like to announce one thing. Okay, I would like to announce one thing to everyone. Okay, that the feedback link will be given anytime during the second second speech. Okay, by the Dr. Bhumik. Yeah. So I hope you all are enjoying the session, and everyone should have to follow the speech, because in the feedback form, if the feedback form in the feedback form there are 10 questions are there, and the 10 questions are from Dr. Dipankar Ghosh and from Dr. Bhumik. Okay, so you all have to, answer all the 10 questions. Okay, and you have to get minimum 40% to get the certificate. So it is my request to everyone, just follow the speech very well, so that you'll be able to answer the question. Okay, and we all know that the time is really tough for everyone, so we are just trying to give you the original scenario of COVID-19 that is going on in recent days. Okay, and so I think we will be able to, yes. So we'll be able to continue our webinar. Yeah, Shreya, we will provide you the feedback link in this chat box. Okay, so just be active, and you have to fill up the chat feedback link. Okay, and I have already told you that there are 10 questions. So you all have to answer. The 10 questions, and you have to get minimum 40% to get the certificate. Okay. So we are really happy to have both the speakers today, and both speakers are very eminent scientists and doctors and professor. Okay, yeah, thank you, Shwati. It's really a good platform because we all are able to know about the recent updates on the COVID-19 therapies and research that are going on throughout India, not only in India but also throughout the world. The last session was also very nice. We become able to understand the molecular basis of all the drugs and the molecular pathways. Okay, sir explained very well. And now Dr. Bhumik, we will start the second part very soon. There are some technical problem, so we are just restarting our system. So just give us 5 minutes and we'll be able to start our second session. Okay. So what I'm telling that Dr. Bhumik is really doing well in every field, not only in medical but also in research field, and he's really great person. So if you have any kind of query, any kind of questions, then you can ask to Dr. Bhumik as well as Dr. Ghosh. And
I think Somnath, sorry to interrupt. I think you are audible in YouTube. Shomo, can you tell me if I am audible as well? Because this looks like a very strange problem. Never happened with me that I'm audible
Yes,
on
sir.
Google Talk. Can you generate? I mean, I don't know. I don't have a solution to this. I'm pretty bad at technical solutions. So, can I just speak? You can record it and then give it in YouTube later. Am I audible, Somnath? Yes, sir.
Yeah, yeah,
What
yeah.
is it?
Yes,
I think
yeah, I can
no.
hear you.
I think Shomo, you are also audible. Okay, great. So
Yeah,
after
yeah, yeah. Okay, okay, sir. You can, yeah, you
finally
can start, sir.
after technical glitch, so we can start. Just great. So
Yes, sir.
thank you.
You
Yeah,
both
thank
are
you.
audible now in YouTube. You can
Okay.
start the session.
Okay, great. So I'll start finally, never better late than never. So,
Thank you, sir.
okay. So, good evening, everyone. As I was mentioning, Dr. Ghosh made my life very, very easy because he has spoken 90% of my slides. And thank you, Shomnath. Thank you, Shomo, for this opportunity. And you people are doing a great job at the Institute of Science, Culture, and Social Studies. Shomnath, great, keep up the good work. Today, my presentation is going to be on what we are witnessing at clinical front. I heard some talk, some portion of the talk from Dr. Ghosh, and that was, I think, was a very technical talk in terms of what is happening in the lab and what could be the drugs, how they could be repurposed. So, just to introduce the topic, novel coronavirus, as we know, is caused by the novel coronavirus 2, which is a novel virulent strain of coronavirus. Coronavirus has been with us for last a few decades. In fact, there have been three epidemics of coronavirus in the last 20 years. And the last one happened in 2012, which is known as a Middle East respiratory syndrome virus epidemic. Why are we so concerned? Because though I was hearing few questions that meant that somebody was asking about asymptomatic infection. Yes, in hospitals at PLS Hospital, we are seeing that 80% of the patients are coming in with no symptoms. In fact, many of our colleagues have been detected positive, but they had no symptoms. So that is one end of the spectrum of the disease. The other end of the spectrum is fatal virulent pneumonia and severe respiratory distress syndrome, which is resulting in lots of death. In fact, the statistics in the world stands close to 7 lakh people have lost their life. So it has been considered a major public health problem and a public health emergency, and the WHO, World Health Organization, has declared this as a pandemic on 12th March 2020. What is pandemic? For majority of the people, I know that it's a heterogeneous group of students who are from different backgrounds of biotechnology, of microbiology, and other biological sciences field. A pandemic is an epidemic which is happening over two continents, more than two continents. So, except for the continent of Antarctica, all other continents are reporting cases. In fact, Latin America, Asia, and Africa are now reporting the large number of cases. So, what are the clinical features? If you look at the published literature, the commonest clinical feature is fever. 88% of the patients come with fever. But having said that, 12% of the patients will not have any fever. So just that, the fever is a diagnostic criteria. Is not that you might have patients who will not have fever. They can just present with dry cough, and there could be a very, very vague fatigue, chronic fatigue. And this fatigue has been described as one of the worst fatigue patients have experienced in the last few years. They can have headache, 14% of the patients. Loss of smell is a very, very characteristic symptom of the disease, which is present in 30% of the population. In fact, in one of the studies reported from Italy, 50% of the patients suffered from loss of smell. Patients can present with shortness of breath, joint pains, and diarrhea. In fact, some of the patients who are having very bad outcomes, they have fever with diarrhea, and they have been called as super spreaders. That means they spread a lot of infection in the society and whoever they come in contact with. So In severe cases, you will have confusion. There would be, because of lack of oxygenation at the level of the lung tissue, patients will have blue face, lips. There could be hemoptysis or coughing of blood, persistent chest pain, and patients eventually will move into kidney failure and will die with multi-organ failure. So it's a very, very dreadful disease. Why do some patients move from one end of the spectrum to the other end of the spectrum? The major pathogenesis is the cytokine storm. So I think my previous speaker spoke in details, and I'm not going to get into details of that. And whenever an antigen like the COVID-19 virus enters into the body, they triggers off immune responses in the form of interleukins, and the macrophages and the dendritic cells. They release a lot of inflammatory cells, which tries to destroy not only the COVID-19 virus but the tissues around the virus. So because the antibodies or the interleukins cannot differentiate between self tissue, which is our own body tissue, and the foreign particle, which is the COVID-19 virus. As a result of which, there will be there is a release of massive number of inflammation. There is massive inflammation, and the inflammation continues to increase because it's a vicious cycle. The virus keeps on replicating, and the inflammation keeps on increasing, resulting in destruction of the lung tissue, alveolar cell, and filling up of the lungs with inflammatory fluid. So if you really see the x-rays of these patients, you will see that the lungs, which are usually black in color because they have air, becomes white in color, and because of the accumulation of the inflammatory fluid in the lungs, and the patients can't breathe. So they require ventilation support. So what is the current management protocol of COVID-19? Currently, there is no cure or vaccines for COVID-19, and that is what is the stand by the World Health Organization and also by the Ministry of Health in India. The present clinical management includes majorly infection prevention and control measures, which also include to do contact tracing and trace whatever whichever contacts have come in whichever contacts have come in the exposure of the patients. We provide supportive care in the form of IV fluids. They are given supplemental oxygen. Some patients require 10 liters per minute. They are given antibiotics. They are given some of the repurposed drugs. Majorly, which is leading the list, is hydroxychloroquine with azithromycin. And finally, if the lungs keep on failing, then the patients are put on ventilator support when they are indicated. So vaccines are actually the other weapon against this deadly virus, and there are multiple of them in the race for development. And the medications which Dr. Ghosh mentioned as repurposed medications are already medications in the market which were used for some other indications are now being investigated in COVID-19. So at PLS Hospital, where I run the clinical research unit, we have currently 5 clinical trials of COVID-19 going on with different products. Some of them are already approved in India for certain disease condition, and some of them are newly being developed for COVID-19. So there's a lot of clinical research work which is happening in hospitals who are interested to do that. This is a huge clinical trial, which is going on by the World Health Organization, which is known as Solidarity Trial. There are over thousands of patients, and hydroxychloroquine is being tried on one arm in combination with azithromycin, and the other arm has ropinavir, ritonavir, and we are trying to look at the outcome. So the results are still not known, and these clinical trials will actually give us information that whether these drugs are acting or not, because whatever data is published in the medical literature, these data is retrospective in nature. That means this is not a prospective randomized controlled trial. And of course, we have a lot of buzz around convalescent plasma therapy. the current status of convalescent plasma therapy in India, that it is still investigational. It is not approved by ICMR, neither by the government of India for the treatment purpose of patients who have got infected with COVID-19. So I already mentioned about the repurposed drugs. Some of the drugs which I would like to mention in clinical, in what are being used clinically is hydroxychloroquine with azithromycin, remdesivir. Favipiravir is a very new molecule which has just been approved by the Indian government 2 weeks back. Tocilizumab is an interleukin 6 monoclonal antibody which is being used in severe infections, and methylprednisolone along with dexamethasone. So these are steroids which calm down the inflammatory cytokine surge. So this, you probably have seen this slide in the last presentation. There are different kinds of drugs who are acting at different levels, starting from hydroxychloroquine, which interferes with glycosylation of the cellular receptor so that the COVID-19 doesn't enter into the host cell. And then we have remdesivir, which is an antiviral drug. It inhibits the viral transcription and produces premature termination of the RNA transcription. And then finally, you also have the convalescent plasma. These are antibodies which are directly blocking the S protein, the spike protein, which is present on the virus, so that they don't bind to the ACE2 receptors present in the lung tissue and other tissues in the body. And then we also have the antiviral drugs which are used in HIV therapy, which is ropinavir and litnavir. They are blocking the polyprotein processing in the viral chain, and especially and resulting in blockage of the viral replication. So few things on hydroxychloroquine. It is primarily an anti-malarial drug, but has shown some positive results in case of the in vitro studies. It primarily inhibits the entry, transport, and post-entry stage of the virus. It increases endosomal pH, so it makes the pH and interferes with the glycosylation, and thereby it has a potential to block the viral infection. The Indian Council of Medical Research, in fact, is the only body in the entire world which has recommended this drug usage not only for treatment but also for prophylaxis for in the healthcare workers. And let me tell you that is only in the healthcare workers. So it is based on the in vitro evidence of hydroxychloroquine does not allow the novel coronavirus to attach to the cells. There are two Indian companies which are manufacturing this drug at very large quantity, Ipca and Zydus Cadila. They are the world's largest manufacturer of the drug. And currently, hydroxychloroquine is used as a standard of care along with azithromycin. Hydroxychloroquine 200 milligram thrice daily is given for the first 5 to 7 days of treatment, along with azithromycin 500 milligram once daily with the patients who are treated in hospitals or in home care. Hydroxychloroquine prophylaxis dosage is 400 milligram twice a day for day one, followed by 400 milligram once weekly for next 7 weeks. And this, of course, cannot be given in patients who have any kind of cardiac problems, especially patients who have QT prolongation and patients who have retinopathy, issues with the eyes. Remdesivir is a drug which has been approved on June 2020, in June 2020 by the Indian company Hetero Drugs, and this has been used. This will be used for restricted emergency use. Remdesivir, let me tell you, has not still undergone any human trials in Indian patients. So these entire data has been extrapolated from the American trial, which showed there was a 30% reduction in hospital stay when these severe patients were put on remdesivir. The medicine has been issued by the US FDA with the emergency use authorization, and the Indian government within couple of weeks has allowed the Indian doctors to use remdesivir in patients with moderate to severe COVID-19 patients. The mechanism of action is it inhibits viral replication through premature termination of RNA transcription. We have other companies like Cipla, Hetero, Jubilant, who are gearing up for the sale in India. Currently in at PLS, we are getting the supply from Hetero Drugs. It's an expensive molecule, 5,400 per vial, and we require close to 20 to 25 vials for the treatment of the patient. So the supply of this drug is also under question because we are not getting a continuous supply of remdesivir for treatment of the patients. Favipiravir is an oral drug. This is the first oral drug which is being used in mild COVID-19 patients, and it's being used it's been marketed in India by Glenmark, which is under the name Faviflu. It's an expensive medication and is costing around 103 rupees per tablet, and a strip costs you 3,500 rupees. And there is a lot of pill load, and that means the patient has to take 18 close to 18 pills, because the first day dosage is 3,600 milligram, which is divided into two different dosage, 1,800 milligram in the morning and 1,800 milligram in the evening, and followed by 800 milligram twice daily for 14 days. So there is a lot of pill load, but this drug is also working wonderfully. The government of Maharashtra, government of Odisha, and the government of Assam has already included it in the government guidelines for the treatment. However, the government of West Bengal is still deliberating whether favipiravir should be used in patients who are with mild to moderate disease. How do this favipiravir has been developed in Japan. It's an oral drug. Let me tell you, all other drugs, hydroxychloroquine is not an antiviral drug. This is an oral antiviral drug, and favipiravir has been tested in not only in Japan but in China. So in China, they had studied in close to 300 patients, 80 in one study and 236 in other studies, and favipiravir treatment led to quicker reduction in viral load and comparison to the other drugs, and early improvement in patient outcome. So the favipiravir has shown better recovery, clinical recovery on day 7 as compared to another antiviral drug, umifenavir. It has resulted in quicker relief from fever and cough. In my practice, I have used favipiravir in some patients, and let me tell you, the drug is really working. By day 4, the fever disappears, and by day 7, the patient gets completely relieved of the cough symptoms. So this drug is very good in mild cases, and patients who are from mild cases are not progressing into moderate cases. Glenmark has conducted a phase 3 clinical trial with favipiravir, whose data has been released last week, and it claims that favipiravir showed clinical improvements of up to 80% in COVID-19 patients, Indian patients, and with rapid reduction of viral load for 4 days. The clinical importance of this is very, very pertinent, because if a patient is staying at home and the viral loads are decreasing, then the chance of infection of the other family members come down significantly. So as a physician, I believe that this drug has a great future, and I think that the government of West Bengal would really include this drug into the standard treatment guidelines, which are still not included in the state. Some words about tocilizumab. Tocilizumab is a monoclonal antibody acting against interleukin-6 receptor and prevents the cytokine storm. And just one update, last week on 29 July, in fact, this week, I would say on 29 July, Roche has reported a negative result with 452 patients in severe COVID-19 patients in Italy, saying that the drug did not achieve the primary outcome of improving the patients on whom tocilizumab was used. It's a very, very important data considering that it's an expensive drug. One vial cost 45,000 rupees, Indian rupees, and you probably have to end up giving 2 to 3 such vials in these patients. They have a lot of adverse events. Tocilizumab can flare up a lot of infections, including latent tuberculosis. It can produce severe suppression of white blood cell count, resulting in secondary bacterial infection in these patients who are usually admitted in ICUs, and a chance of hospital acquired infection is very high. It can bring about fungal infections. So the question here is, with the result, a negative result coming in, which is a very, very recent update from patients in Europe. Do we really justify the use of this drug in the Indian context? That is something which we need to think about. Corticosteroids, one steroid has actually taken the entire focus on itself, is dexamethasone, which is a very, very cheap steroid, which is available for 20-30 rupees a strip in India. And the huge trial was done on 1,000 patients in Oxford University. The name of the trial is Recovery Trial. It shows that dexamethasone produced 30% reduction in mortality. I personally believe that this is a huge, huge data which is coming in very recently, and we need to be careful about it. And corticosteroids, they are anti-inflammatory, they are immunomodulatory drugs. So the major pathogenesis which happens in a patient with moderate to severe disease is cytokine storm, is massive inflammation of the lung tissue, is this accumulation of the inflammatory fluid. So the steroids become extremely important in the management of these patients, and Indian doctors use the long-acting steroid, which is methylprednisolone. The methylprednisolone has been used in medicine for a very, very long time. They're used in rheumatoid arthritis, they're used in bronchial asthma, inflammation in COPD, and methylprednisolone in our own hospital also. Our doctors are using methylprednisolone to to reduce the flare of this disease and reduce the cytokine storms. So this is already published, the data of the efficacy of methylprednisolone, and of course dexamethasone data is undergoing publication in a decent journal. So corticosteroids is another drug which is being used in the treatment of COVID-19 moderate to severe patients. Convalescent plasma, I think it is very early days to speak something about it. The most important thing is that it is still not an approved therapy. As I mentioned, it binds on, it's an antibody which is the IgG antibody which is taken out from the plasma, and this IgG antibody binds with the spike protein so that it doesn't interact with the receptors present in the human cells, and resulting in decrease in the viral concentration in the human cell. That the most important thing is that these are undergoing clinical trials in India, so it will be too immature and premature to say that convalescent therapy is working. But in a pandemic situation, we have to understand that every therapy what we use is quote unquote experimental on patients, and every particular drug and every particular therapy, including convalescent plasma, requires informed consent. That means the patients are told, if not the patients are able to understand, the family members are told and counseled that this therapy might work, might not work, but because there is nothing available right now at our hand, we are going to use this with the intention that this will work. So people have to sign off a document known as the informed consent document, and the government ensures that we take this informed consent from every patient who will be treated with these experimental therapy. Ropinavir and Litnavir are two antiviral drugs. They prevent the polyprotein production, and they are by cleaving it into functional unit. So it inhibits the proteolysis. The drug is already approved in Russia. So a country like Russia has already approved Ropinavir, Ritonavir as the first drug for treatment of patients who are coming in with COVID-19. Russia is also seeing massive number of cases, and ropinavir, litnavir, by the recommendation from the Indian government is that it should be used in patients where hydroxychloroquine could not be used. One of the major complications, I said, of hydroxychloroquine is that it cannot be used in patients who have cardiac problems, who are on pacemakers, who have major eye problems, including the retinopathy, myopic retinopathy. So these patients are put on ritonavir and and lopinavir in the hospitals as well. So a few updates on the vaccines, and there is a huge race on in the world that which company or which country will bring the vaccines. Currently, there are more than 150 vaccine candidates which are in various stages of trial for coronavirus. And the WHO experts have said that the first use cannot be expected till early 2021. So if somebody is expecting that the vaccine will come in November, December, or on 15th August, let me tell you that I have to dishearten you. That is not going to happen. These vaccine trials follow a certain methodology. Vaccine trials follow a certain protocol. Safety is extremely important in vaccine trials. It should not happen that the vaccine comes into the market and the patients get infection from the vaccine. This is from the WHO website, that what is the current status of the vaccines, and I thought this is a very, very comprehensive list. Leading the list is Moderna Inc. from the United States. It has got a huge support from the NIH, National Institute of Health in the United States, and the large phase 3 trial has begun on July 27th. And it is planning that trial is going to happen over more than 250 sites in the United States. It's an RNA vaccine, and US is leading the list. Closely followed by the United Kingdom, where Oxford University has already published the successful phase 2 clinical trial data in 1,077 patients, and Oxford has collaborated with AstraZeneca on one hand, and AstraZeneca has done a collaboration with Serum Institute of India, which is also developing the vaccine in India. Serum Institute, in fact, is developing the vaccine as simultaneously as the clinical trials will go on. And so the trials are expected to begin in August 2020. But the vaccine development is already going on in Serum Institute factory, with the aim that if the positive results come in, the world population should be able to receive the vaccine. It's based on a non-replicating virus known as the Shadox virus. It is extracted from chimpanzee, so it's known as the Shadox vaccine. And the entire work is happening at the Oxford University in the United Kingdom. India is not also lagging behind. India's own Bharat Biotech, Hyderabad-based company, is developing Covaxin. It's the phase 1, phase 2 data trials have started. It's on inactivated virus, and ICMR is supporting Covaxin development. You can see Russia has come up with 2 important vaccines. One of them is actually military vaccine. And I must mention the name of China here, where CanSino Biologics, which is a Chinese company, is in neck-to-neck race with Moderna Inc. in the United States, because that has also entered into the phase 3 trial. So we have to understand, and then you have, of course, the other countries like Australia and Canada, which are following the different vaccine. But I think the top 5 which are mentioned here are the one where the entire competition is revolving around. So just few words before I end my presentation on why so much time will be required, because you have to understand that vaccine trials happen through phases, and each phase is important. So if you read the newspapers, or you if you understand, or you go through the televisions which keep on saying that a successful vaccine has been reported, please understand which phases the development is going through. The phase one primarily is focused on safety and immunogenicity, so that we want to understand that when we inject the vaccine, there are no complications in healthy volunteers. Phase two is also safety and dose ranging study. So we want to find out whether we want to give a single dose or a double dose, and this has a direct correlation with immunogenicity. And phase three is the most important part of vaccine development, is a large scale safety and efficacy study, which will happen in thousands of patients. So majority of the data which is being reported in the media is on basis of this phase two vaccine reports, which are coming in. It might happen that in phase three, the vaccines might fail. So I can give you a statistics that on an average, a proper vaccine development requires 7 to 10 years. And we are trying to hasten up. We are trying to fasten it up. And let me tell you that there is nothing called that, you have to develop a COVID-19 timeline vaccine or a non-COVID-19 timeline vaccine. Every phase is important, and every phase requires certain commitment for patient safety, certain commitment for efficacy. So there are in history, there are instances where the vaccine has worked in limited hundreds of patients of phase two, but when it was tested in thousands of patients in phase three, the vaccine failed to generate any efficacy. So as I mentioned, it is very, very early days to rejoice that the vaccine will come very soon. So, and there are some ethical considerations which are also done, because vaccine trials don't happen in silos. We have to follow what is known as a good clinical practice. I am not going to get into details. Maybe Shomnath can organize one session on good clinical practice and clinical research going ahead, and we can train people on what good clinical practice is all about. Every institution where this clinical trial will happen, these institution has to approve this particular clinical trial through its institutional ethics committee. I am the member secretary of the PLS's institutional ethics committee, and I'm attached to 4 such ethics committees and 4 hospitals in Kolkata. Let me tell you that institutional ethics committees consist of non-medical people more than the medical people. So ethics is a very important component. Who are you going to give the vaccine? Why that person is going to receive the vaccine? Just because the person requires the vaccine, are you giving it? Or is the person vulnerable? Is the person the housekeeping person who's working in your hospital whom you're going to inject the vaccine? All these ethical issues have to be taken into consideration. Informed consent has to be taken. You inform everything about the vaccine, what are the positive events as well as what are the adverse events. We always have to talk about what can go wrong. And we have to also ensure that there is no exposure to unreasonable risk. And if there is exposure to unreasonable risk, then the entire care of the patients have to be taken care of when the trials are going on. The economically and socially derived communities should not be exploited, a very common feature in developing countries like India. So we have to be very, very careful that, in the course of being over enthusiastic of developing a vaccine, we should not inject a vaccine into these communities and exploit them. And of course, what would be the benefit of including this vaccine into the national immunization program. I've already discussed this. The phase 1 is usually in very less number of patients. Just see 100 patients. They're open label. That means everybody knows everything. Doctor knows when he's injecting the vaccine. The patient knows that, or the healthy volunteer knows. And they are not randomized with placebo. Placebo is any pharmacological ingredient which does not have any activity. So we give this to reduce the bias in the clinical trial, and we need a good adequate laboratory support and proper monitoring is done. So this is very, very important that the safety is tested in phase 1. Phase 2, several hundreds of patients, can be usually 1,000 thousands of high risk subjects. In fact, we just did one BCG vaccine trial for NICED in PLS, where we have tested that BCG vaccine in healthcare workers. Does BCG reboosting second dose increases fight with coronavirus? That is what we have studied. And so this usually happens, and this study happened in over 25 sites, and PLS was one of the sites from Eastern India. And they are randomized, well-controlled trials where every age, ethnicity, gender, and genotypes are included. Phase 3 is a huge study, and what we look at is we usually look at active monitoring. We for live attenuated vaccine, some of them could be live attenuated vaccine. The monitoring can be done till the second and third week. That because whether the vaccine itself has produced the disease or not, and then the groups are divided as responders and non-responders, and this is how we calculate the goal. The goal is now the goal has changed to efficacy. So if you see, when you started phase 1, the focus was on safety. Now the goal has changed to efficacy in phase 3. So if the efficacy does not happen, then it will be a failure of the vaccine. And the vaccine efficacy is calculated with the help of this formula. A few updates, as I mentioned, on Covaxin. Being an Indian, I think I'm very, very enthusiastic to look at what this Bharat Biotech's vaccine with ICMR is going to bring about. 12 sites have already started phase 1 study because ICMR is collaborating with Bharat Biotech for this Covaxin corona vaccine. AIIMS New Delhi has started the phase 1 trial last week. The first patient has already been dosed. And initial reports from PGIMS, Postgraduate Institute of Medical Sciences in Rohtak in Haryana, has shown positive phase 1 results. The phase 1 will finish and will progress to phase 2 to demonstrate safety and immune response. The CanSino, as I mentioned, is another very, very prospective vaccine candidate from China. It's being developed in China. It's already completed the phase 2 phase, and there is, they are trying multiple booster shots. They are trying to give two booster shots here, and this has already started. This is on the verge of starting the phase 3 trials in Canada. So CanSino is another very, very prospective vaccine candidate. Our focus now shifts on Moderna NIH vaccine, and Moderna is a biotechnology company based out of United States in Virginia, and they have already started the large phase 3 trial in USA on July 27. Over 250 sites with 30,000 participants are going to be enrolled, irrespective of the age, sex, and ethnicity. So everybody is going to be involved. Vaccine already has got a huge support from the US government, and a funding of $1 billion have already been given to this company to develop the vaccine. Results of this phase 3 trials are expected by November 2020, and if the FDA facilitates an expedited approval, then by December 2020 the vaccine might see the ray of light. Oxford AstraZeneca serum vaccine collaboration, positive results of phase 2 has been reported in Lancet. Over 1,077 healthy volunteers were involved. Serum Institute of India. India is also playing an important part through Serum Institute and has collaborated with AstraZeneca. And this is one of the first of its kind in modern history that simultaneous production of the vaccine is going on. So if you remember, Serum Institute of India is already producing this vaccine in its factories with the hope that the vaccine will be successful in the phase 3. Phase 3 is about to begin in the first week of August, this week or next week, because the DCGI is about to give the approval any day, and the results are expected by December 2020. There's a third vaccine, which is the Pfizer biotech coronavirus vaccine. The US drug major Pfizer has collaborated with the German company Biotech, which has already done the phase 1 study. And the Pfizer is hoping to get a lot of the US FDA support for early approval of the vaccine as early as October. But I think the vaccine will not come to the market before December. So we, as I mentioned in my previous slides, all the phases have to be completed, and all the phases have to be ensured that the vaccine becomes a safe vaccine. So to end my presentation, I would say that COVID-19 is a new disease, and the information about it is revolving. Whatever I presented today, if I come back after a month, the slides will be totally different. So you have to keep yourself updated, keep reading the medical literature, keep reading the literature which is being published on a regular basis. And let me tell you that majority of the literature is free, is currently free on the web. No journal is charging you for downloading those articles. There is no definitive curative medicine for the disease. So if you are thinking that the drugs which I showed, favipiravir, remdesivir, ritonavir, lopinavir, hydroxychloroquine, I must dishearten you to tell you that this is not a definitive curative medication, because we still don't have large randomized controlled trials to prove that the medication is working. Repurposed drugs are currently being used, which to top the list are hydroxychloroquine, remdesivir, tocilizumab. I don't know how long tocilizumab will be used, because only 2 days back, a very, very negative data has come on tocilizumab in European patients. Large clinical trials on the efficacy of these drugs are awaited. Over 150 vaccines are currently in development, and Moderna NIH vaccine from the United States and CanSino from China is ahead in race, because both are about to start the phase 3 trial. So we're keeping our fingers crossed, and we are going to observe how these two vaccines are going to fare in phase 3. So to end, I would say prevention of the disease is the best strategy by following the SMS strategy. Please maintain social distancing. S, wear the mask, the current kind of mask. Don't wear those cloth masks. I'll make a request through this platform. Wear the surgical mask, because cloth masks are equivalent to no mask at all. If you sweat and your mask is wet, then you are actually moving around with no mask. So wear the right kind of mask, and regular hand sanitization. SMS is the only strategy which we have to follow in the current context, and we have to really keep our fingers crossed that very soon a vaccine, or very soon a curative drug, will evolve. With this, I would like to end my presentation. If there are any questions, I'm more than happy to answer them. I'll definitely try to answer.
Thank you, Dr. Bhumik. Yes, so now Shomu, please ask the questions, okay, that are given in the chat box.
Yes, sir. So we have several questions in the chat box, starting with Shubham Shah asked, as the virus has high mutation rate, is it possible, is it fruitful to target spike proteins, or rather viral nucleic acid targeting for vaccine or drug will be fruitful more?
No, I think the mutation rate is just like the influenza virus, because as they come from both the same families. So definitely vaccine is going to be effective, because we are going to target the serotype which causes majority of the infection. So yes, vaccines are going to be effective in development. Drugs are also undergoing, a lot of drugs are undergoing development. So it has to be a combination approach. If you look at the treatment of influenza, it's not that the vaccines are only used in the treatment of influenza. Majority of them are treated by the drugs like oseltamivir. So you have neuraminidase inhibitors, which are also playing a very important role. So just because a virus mutates, we cannot stop the vaccine development. It has to be a collaborative approach of drugs plus vaccine for saving the human population.
So going to the next question asked by Devoshmita Banerjee. Can inhibition of double membrane vesicle formation, within which the replication of viral genome is occurring, be a target for COVID-19 therapy?
I think that is definitely possible. I am not a biotechnologist and I will not be able to answer that question. So I think maybe Dr. Ghosh is the right person to answer that question. But I would say any stage of viral replication, if that is inhibited, that could definitely be a target for the drug development. The only question is that we have to understand that every drug development requires a huge amount of funding, and we spend close to 1.5 to 2 billion US dollars to develop a drug, and there are usually on an average 10 to 12 years spent. So question here is that if that is a feasible option or not, that we can find out through high throughput screening, which are now readily available with the incident, with the development of artificial intelligence. And the high throughput screening can actually develop models that whether this can be feasible or not. So I think this has to be this has to be checked first in the laboratory and then in the in vitro models, and following which we have to progress to the clinical trials. So we have to understand the complexity of this development, and the failure can come in every phase. Let me tell the audience that out of 10,000 drugs which enter in phase 1, this is a startling statistics. Out of 10,000 drugs which enter into phase 1, only one will see the regulatory approval. So the failure rate is as high as 99%. So there's a high incidence of drug failure rates. So we have to really understand economically and commercially, as well as biochemically, whether that is possible or not. That decision has to be taken by a team and not by an individual.
Going to the next question, Shauradip Mullik asked if the virulence of COVID-19 decreases later on and the vaccines which are in clinical trial now, will they produce cytokine storm when injected to the patient, and what will be the consequences?
No, I don't think so. The vaccines will produce cytokine storm because it will only do, because majority of the vaccines are dosed in a way that they only generate an immune response. But having said that, the possibility of cytokine storms have to be ruled out through vaccine vigilance program. So let me again add this. What is this vaccine vigilance program? Even if the vaccine is approved, so safety development of the vaccine starts from the day 1 of phase 1, goes on till phase 2, phase 3. And let me tell you that it does not finish there. It also continues when the vaccine comes into the market. So when a regulator, the Indian regulator will give somebody an approval status, the vaccine has regular information about the safety data of the vaccine has to be submitted by the company which has got the license which has got the license to market. That is known as the vaccine vigilance program. So if the vaccine vigilance program throws up that yes, there are chances of cytokine release syndrome happening in patients who are receiving the vaccine, the vaccines will be withdrawn. That has happened with oral polio vaccines. Some of the oral polio vaccines have resulted in vaccine-induced paralysis, which were later withdrawn from the market. So the safety reporting and the safety vigilance continues throughout the life cycle of vaccine when it is in development, as well as when the vaccine comes into the market. It is not a stop process when the moment the drug comes into the vaccine comes into the market.
So this is the last question asked by Neil Somudro. Can phase 1 and phase 2 of vaccine trial be clubbed together?
Yes, that's a very good question. So yes, they can be clubbed together. But we have to understand that adequate sample size have to reach for both the phase 1 and phase 2. In COVID-19 pandemic, we are clubbing these. That is true, and that can be done. Yes, in case of emergency situation, they can be clubbed together.
Sir, one question from my side. What is your say on the Russian vaccine who are claiming that they will be approving it by August 10?
I think everything is very premature and immature. It is just like somebody saying that in India vaccine will come on 15th August. I think a lot of the media is being misquoted. So we should just refrain from such information. As I mentioned, that this will require a lot of monitoring. And if the publication comes out in peer-reviewed, in peer-reviewed journals, medical journals, then only we should accept it. And last but certainly not the least, if WHO endorses, everything said and done, the global body which controls healthcare policies in the world is World Health Organization. So until and unless World Health Organization endorses a vaccine or a definitive drug as the curative cure, nobody, no person on earth can claim that the vaccine has arrived or the vaccine is already, we have the vaccine. So everything will be under subject of evaluation from the World Health Organization, and World Health Organization, once it endorses, we should have that information with us.
Okay. Thank you, sir.
Okay. Thank you so much, Dr. Bhumik and Shomo. So here we are at the end of the webinar, and we really thank to Dr. Bhumik and Dr. Ghosh both for giving their precious time to us.
We should thank you.
Thank
We
you
should
so
thank
much,
you
sir.
first.
Yeah, thank you
Thank
very
you.
much for the opportunity.
Thank you so much. It's my pleasure to have you both. Okay, sir. For everyone, I would like to conclude uh, by saying just one thing. Just subscribe our channel, YouTube channel, so that you all will you all will be able to get the information about the next upcoming webinar series that will be for both national level and international level. And we also would like to tell you that we are organizing different national level competition. For now, the art competition and the dance competition is going on. So if you would like to participate, you can. The date is extended up to 10th of August. And lastly, finally, I would like to thank all the members in the organizing committee, Dr. Dipankar Ghosh, Dr. Subhrajyoti Bhumik, Shomo, Priyanka, and all the technical staffs. Thank you so much, and thank you all. So I'm concluding this webinar here.
Thank you.
Thank
Stay
you so much,
safe.
sir.
Thank you. Good night. Good
Stay
night.
safe. Thank you.
Thank you,
Good
sir.
night. Bye-bye.
Good
Bye.
night, sir. Bye, sir.